day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Probe-Based qPCR Approaches for Detection of Single Nucleotide Polymorphisms and Validation of Molecular Diagnostics. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for approximately 90 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using that chat panel. And at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically for you. Please note that this event will be recorded and made available for future download. And at this point, I would like to thank BioSearch Technologies, who helped develop the content for this presentation. BioSearch Technologies is a privately held ISO 9001 and ISO 13485 certified company located in Northern California. Founded in 1993, BioSearch is a leader in the design, development, and manufacture of sophisticated oligonucleotides for real-time qPCR application. BioSearch produces a variety of probe formats, including dual-labeled BHQ probes, BHQplus probes, molecular beacons, and scorpions primers. More recently, BioSearch started offering value mix assays for Snip genotyping, which comprises custom BHQplus probes and primers combined with a single tube. This formulation minimizes pipetting error and simplifies your protocol by providing mixed oligonucleotides ready for Snip genotyping. BioSearch supplies businesses with custom oligos for all stages of product development, from design through commercialization. A vertically integrated structure allows BioSearch to precisely control the raw materials and manufacturing process, reducing costs and turnaround time. BioSearch's markets include pharmaceutical, in vitro diagnostics, biotechnology, AG-bio, environmental, and food testing, public health, and biodefense sectors. And now, I'd like to introduce our first speaker for today's event. James Willie is Professor of Medicine and Pathology and George Isaac Professor for Cancer Research at the University of Toledo Health Sciences campus in Ohio, as well as co-founder and consultant for AccuGenomics. Professor Willie is holder of more than 10 U.S. patents in molecular diagnostic tests and methods. The current focus of research includes development of molecular diagnostic assays, novel quality-controlled target next-generation sequencing methods, and novel approaches to understanding complex genetic traits. Professor Willie was the corresponding author on a recent paper describing a novel two-color fluorometric RT-qPCR method that provides better quality control for diagnostic testing. This method is licensed to AccuGenomics. And our second speaker for today, Cassie Temple, is the Laboratory Operations Manager at Douglas Scientific in Alexandria, Minnesota. She grew up in Alexandria and attended St. Olaf College, in where she majored in chemistry. She completed her Master of Science degree in Food Science and Human Nutrition at Iowa State University. Upon graduation, Cassie worked in genetic research in the Department of Pediatrics at the University of Iowa. When her family moved to St. Louis, she continued her career at the Washington University and St. Louis School of Medicine. There she spent 8 years working in genetics and molecular biology research in the fields of heritable bone deformities and oncology. She joined Douglas Scientific in the summer of 2012. And now, without further ado, I would like to hand the mic over to Dr. James Willie. Dr. Willie, you can begin your presentation when ready.
Thank you very much, Andrew. I'm going to minimize this window, and very good. Okay, so thank you very much for that introduction. I did modify the title of my presentation slightly to say use of BHQplus probes to optimize multiplex competitive quantitative real-time PCR and diagnostics. And I just mentioned that I am a Chief Medical Advisor for Accugenomics Incorporated in Wilmington, and I do have equity interest in that company. And funding for much of this, what I'm presenting today, came from NIH through the grants that are mentioned down below. Okay, so the outline is I'm first going to speak briefly about the basics of competitive quantitative real-time PCR, advantages of this approach in discovery and diagnostics, and I'll mention applications that are currently in commercially available diagnostic tests through Roche for infectious microbes. Then I'll go on to describe the applications of multiplex competitive RT-PCR and test design, talking about the multiplex preamplification with a mixture of internal standards, which is a technology that is licensed to Accugenomics, and followed by a second round amplification with BHQplus probes. Then I'm going to describe specific applications in diagnostics of the multiplex competitive real-time PCR approach. First, in the lung cancer diagnostic test, or LCDT, that we developed here, that will be used in diagnosis of formalin-fixed paraffin-embedded samples. I'm also going to describe a test we developed in collaboration with the Lake Erie Center, which is a viral hemorrhagic septicemia test for diagnosis of this disease in fish, which is a potential problem, an epidemic problem in the Great Lake. So the advantages of competitive quantitative PCR, basically in each PCR, a target native template is amplified in competition with a known number of respective synthetic internal standard molecules, and the native template and the internal standard compete for the same primers, and this provides for quality control for any kind of suboptimal condition in the PCR reaction, such as interfering substances in the samples or suboptimal reagents in terms of quality or quantity of the reagents in the reaction. Very basically, the experiment setup is that each sample is mixed with a known concentration of internal standard. This fixes the concentration of the transcript target native template relative to its respective internal standard template. Then aliquot a portion of this sample internal standard mixture into each well containing the PCR reagents and primers, and the primers will then amplify the native template and internal standard in competition. The idea is that because they are amplified with the same efficiency, that the starting relative concentration of native template and internal standard will be the same relative portion at the end of PCR, so that signal will represent what was present at the beginning. The advantages of this approach are that it provides absolute quantification without a need for an external standard curve, and it controls for interfering substances and false negatives, and because each reaction has that competitive internal standard in it. So briefly, the basic type of competitive PCR, some approaches, some tests that have been approved by the FDA include the tests for infectious microbes, such as the Roche Probaste AmpliScreen HIV test. This is a test where a single analyte, a particular HIV-1 gene, is measured relative to a known number of synthetic internal standard molecules, and this has been on the market for several years. Now I'm going to describe some of the challenges that and the opportunities for the more complex measurement of many transcript abundance of multiple analytes, which is required for many different types of transcript abundance-based tests. So many transcript abundance-based tests require measurement of at least 2 analytes, and that includes at least one reference gene, such as beta-actin or GAPDH, or several others that serve as a loading control reference gene, and at least one target gene. And then each of these reference genes and target genes is measured relative to a respective internal standard. So for each of these analytes that's being measured, one measures them relative to a known number of internal standard molecules. So a solution to this type of approach is to make a mixture of synthetic internal standards, and this type of approach is covered by a patent 2 Genomics described here. And then this mixture of internal standards is combined with a sample, and each analyte in that sample, each of the multiple different analytes and its respective internal standard, is amplified in a multiplex competitive RT-PCR reaction. So a second challenge that I'm going to get into, and I think we have solutions to that I'm going to describe today, is that most, I'm going to describe a test for diagnosis of lung cancer. And lung cancer is the most common cancer cause of death. It kills more than the next 3 cancers combined, including colon, breast, and prostate. And a reason why it kills so many people is that it's not symptomatic until late in the disease. So 85% of lung cancers are diagnosed late when they're not really amenable to surgery. So as a consequence, the attempt is to make the biopsy as minimally invasive as possible. So it's typically through a fine needle aspirate, which provides a very small sample. And then the second thing that happens is that fine needle aspirate goes into formalin fixation and paraffin embedding for cytomorphologic analysis, which is the main way that each of these tumors is diagnosed. But this is not an optimal approach for molecular diagnostics because formalin fixation and paraffin embedding damages the RNA and the DNA and reduces the RT and PCR efficiency by as much as 100-fold. So solutions that we're using then are, we would use this multiplex method to assess many targets simultaneously so that reduces the amount of sample that's required. So do a first-round multiplex of the sample mixed with the mixture of internal standards, and this conserves the sample and amplifies the signal. And then we do a second-round PCR with a single primer pair and native template and internal standard probes, the BHQplus probes. So I'm going to go through all of this. So the outline then is we developed a standardized 2-color fluorometric assay, and I'm going to describe 2 different tests, the 4-gene lung cancer diagnostic test, or LCDT, which comprises, in this case, 4 different analytes, the E2F1 and MYC genes, which tend to be upregulated in lung cancer, and the CDKN1A gene, which tends to put the brakes on cell proliferation and tends to be downregulated in lung cancer, and then a beta-actin ACTB gene, which serves as the loading control. This test was developed in our lab as many as 10 years ago, described in this article by Warner et al., and then converted to the 2-color fluorometric approach in 2014 in this PLOS One article by Ya et al., which is accessible through PLOS One. The second test I'm going to describe is a 2-color fluorometric assay for the fish viral hemorrhagic septicemia virus, and that was in Pierce et al. In PLOS One in 2013. So the first step then is to prepare the reagents, including the synthetic internal standards for each gene, and then prepare a mixture of internal standards, and prepare a FAM probe for each native template, FAM BHQplus probe for each native template, and a Quasar 670 BHQplus probe for each internal standard. Then I'm going to go through the how we conduct the analytical validation experiments for each test, and then show the results of evaluating these assays and diagnostic samples. So I'm going to skip and then come back to that. So first I'm going to show how we develop the test for each assay. We go to a primer design pathway, such as the real-time design software at BioSearch or Oligo 6, identify the optimal primer binding sites for efficiency based on the in silico analysis for the particular assay. And now after we design those primers, then we do testing to ensure that they are efficient, because although the software is useful, it is always important to confirm the efficiency of these primers in actual experiments. Once the efficiency of the primers is confirmed, then we develop a probe for the location between the primers, and we have a FAM probe that we develop that's perfectly homologous to the native template sequence. As this, after this probe binds, then the polymerase comes through and causes the breakup of the probe so that the fluor is separated from the quencher and the signal is then observed. We also develop a probe for the internal standard. We develop an internal standard that has 4 to 6 base pairs difference from the native template, and then develop a probe, a Quasar 670 probe that's specificity for the SAM probe to the native template and specificity of the Quasar 670 probe to the internal standard. This is just demonstrating how these amplify with the same efficiency, with the green as the native template and the Quasar 670 as the internal standard. We can dilute this sequentially over 6 orders of magnitude and still see the efficiency is the same between the native template and internal standard. So to summarize, the internal standard is designed with 4 to 6 base pairs different for the native template with a Quasar 670 probe and the native template with the SAM probe. The native template and internal standard then compete for the same primers and amplify with the same efficiency as observed here. And we can then quantify the amount of native template that was present in the reaction by comparing the threshold, the CQ for the native template relative to the CQ for the internal standard, and that proportional difference is going to be then compared to the amount of internal standard that we loaded at the beginning, and I'll go through that again on another slide. So I wanted to come back then to this slide to show this is how we quantify the internal standard. We use a digital Pcr quantification as it's commonly used for quantifying internal standards. Our products tend to be 60 to 80 base pairs long. We do a limiting dilution, and this is the same. We diluted from at 10 different dilution points, starting at 40 molecules per reaction down to 0.1 molecules per reaction. And with that, based on photon distribution and stochastic sampling, we should see a reducing number of positive values compared to the observed compared to the expected. And this does, in fact, confirm that the concentration that we thought we had is at that concentration. So this is how we confirm the concentration of the internal standard. Then this is analytical validation. I have a few of these slides to show linearity, confirming that the observed to expected E12-1 values by the fluorometric are as we expected. So this is E12-1, one of the assays in the TAP showing the linearity. This is the linearity of E12-1 over 6 orders of magnitude. And this is just diluting the internal standard and the native template down over this order of magnitude, showing that they're in parallel. And then we also dilute the native template relative to the internal standard and the internal standard relative to the native template to demonstrate that we have linearity in that way as well. One of the advantages of making the 4 to 6 base pairs different in the probe compared to the native template is that we do have very high specificity, and so we can see this linearity over this very wide range. The advantage of this then is that we only need 2 internal standard mixtures to cover nearly 8 logs of measurement, which is perfectly adequate for any gene expression measurements or even measurement of rare early diagnosis of cancer like BCR-ABL in circulating blood. So, and then this is assessment of precision and sensitivity. We can dilute down to 6 molecules in the reaction. Now, as you go down to that level, then stochastic sampling begins to have an effect. The CC goes up at that level. So we are able to reliably measure down to as few as 10 molecules. That, as I say, the limitation is only the stochastic sampling that begins to become evident at these lower levels. And this is another indication of the precision and the CD for 4 replicates at each dilution here of the native template is relative to IS is down, and we can see even down to measurements of 1 to 80 of native templates internal standard. We have very good CDs, but it does begin to become higher as you go down to the greater dilution. So to finalize how we quantify the copy number for each gene native template in the cDNA sample by calculating the native template CQ minus the internal standard CQ for the unknown sample. We then multiply the 2 to the minus delta CQ times the input internal standard molecule copies in the reaction, and that gives us the native template copy number. We then normalize the target gene value to the loaded control gene value, such as beta-actin, and that gives us a final readout of target gene NP molecules to 10 to the 6th reference gene, in this case beta-actin molecule. Now, one more aspect of our measurement is that it is possible that the fluor could vary from experiment to experiment in terms of its signal, or the software may select different CQ values depending on its experimental conditions. So we have something called an external standard mixture that controls for that. Oh, I'm sorry. This slide, before I get to that, is demonstrating robustness, and it's showing that when we put in different amounts of an interfering substance, in this case EDTA, that the internal standards control for that. So you can see as we increase the EDTA concentration that it has some effect on the CQ on the threshold cycle for beta-actin. It begins to have an effect on that before it does on the MIC, and eventually it drops out altogether, and the beta-actin values amplification drops out before the MIC does. So to control for this, we have the internal standards which do control for that value, and you do get a reliable measurement. And then when the reaction stops altogether, we don't read out any kind of value for MIC or the beta-actin because the internal standard is not working. So we always have that quality control with the internal standard. And that's demonstrated here, although these values are beginning to drift for both the internal standard and native template for beta-actin, they level in terms of when you compare the native template to the internal standard and get that value here. And then out here, when you have very high levels of interference, you just don't get any signal. And we would not say, in this instance, for example, that there's no MIC there. We would say that the test didn't work because of interference. So that quality control is very important for each kind of diagnostic measurement. And then this is another form of robustness to correct for a variation in the fluorometric intensity. This is an external standards mixture in which there's a known one-to-one concentration of each synthetic native template and each synthetic internal standard. And this external standards mixture is the same one that's used in each experiment. It stays stable across time. And so it corrects for variation in experimental conditions, including fluorescent-specific activity, such as may happen if from lot to lot or with freeze-thaw, that the labeled probe relative to the total probe may go down. So it controls for that. And then also we and others have probably observed that the CQ selection, the software may select different CQ values depending on the experimental conditions, and this approach controls for that as well. So the sample NT CQ minus internal standard CQ values are then corrected relative to the external standard mixture NT CQ and internal standard CQ. So as you can see here, if we intentionally dilute the labeled NT probe with a unlabeled material so that the specific activity of fluor goes down, then you can see that the quantitative cycle, the quantification cycle for the MIC in this case would go up, but for the NT, because we are diluting the NT probe with the unlabeled probe, so the CQ goes up. But if we control for that by having the external standard mixture, then you can see that this is controlled here. And so this is an important correction factor. So the final then correction, the final quantification is that we calculate the NT-CQ minus the internal standard CQ for the unknown sample, and then we calculate the average NT-CQ minus internal standard CQ for the external standard mixture at two different concentrations, and then we calculate the corrected delta CQ as the NT-CQ minus internal standard QC for the sample minus the NT-CQ minus internal standard CQ for the external standard mixture, and then multiply 2 to the minus corrected delta CQ value times the internal standard copies in the reaction, and that gives us the NT copy. So, and then we normalize against the beta-actin reference gene value. So the summary of test development then for each test is we develop a synthetic internal standard for each gene, and that controls for interfering substances in the PCR, prevents false negatives. We make a mixture of internal standards for each of the targets, and that controls for pipetting variation and other factors. We develop a BHQplus probe, a FAM label for each NT probe, and a Quasar 670 probe for each internal standard. We go through the analytical validation process, ensure that the internal standard concentration is correct to the digital PCR, and then do kind of assess the linearity, assess the lower detection threshold, the imprecision, and we confirm that the external standard mixture is controlling for lot-to-lot variation, experimental variation cycle threshold. So next I want to show the application of these tests. The first one is application of the LTDT to analysis of surgical FFPE lung samples. The opportunity is that there are large archives of these formalin-fixed paraffin-embedded samples from subjects with known outcomes and response to specific treatment. And also there are fine needle aspirate samples which is the most common method for diagnosing lung cancer. And as I mentioned, the problem is that there's a poor quality RNA from these FFPE and FNA cell block samples. The challenge is to develop RT-PCR methods that reliably measure these samples in which the RNA is contaminated with interfering substances, highly degraded, and so the analytical platform must be able to analyze these small degraded samples in these small PCR products. So at this point, we have a poll question to assess the viewing audience as to which type of samples people are using. So I'm going to give over to that poll organizer.
Great. Well, thank you for that. We certainly do have the polling question appearing on the screens of the audience members right now. That question again, what type of samples are you conducting qRT-PCR testing in? Your options are biopsy, surgical fresh frozen, biopsy, surgical FFPE, another option, mammalian cell culture, and your final option there, bacterial, fungal, or viral culture. And you can actually select more than one answer here if you're working with more than one type of sample. So it looks like most of you have clicked and voted for your answers. I'm going to close polling now so you can get your last clicks in because I'm going to share the results with you now. And there we have it. The most popular answer was biopsy surgical FFPE. That was followed by a bacterial, fungal, or viral culture. That was followed again by mammalian cell culture, and the least popular answer was biopsy surgical fresh frozen. And with that, Dr. Willie, I'll hand back the mic to you.
Okay, great, thank you. So let's see, I do have a blackout on the bottom part of my screen, Andrew. Let's see what I can do about that. I'm going to, okay. Okay, I think it's come back, okay. All right, very good. So, so the next slide then is, let me just move this over. Okay, so again, just another schematic. So for processing fine needle aspirate samples, most of the needle aspirate is injected onto a slide for cytomorphic analysis, and that the challenge here is that for cytomorphologic analysis, it's only 75 to 85% accurate. This varies from one pathology lab to another, but then the remaining cells, after putting cells on the slide for morphologic analysis, a small amount goes into the cell block, and that is also processed through formalin fixation for cell block. So the opportunity that we saw is to augment cytomorphologic diagnosis accuracy using a molecular diagnostic analysis test. And the challenge is that to reliably measure DNA or RNA targets in small degraded samples. And the solution was to develop a multiplex competitive RT-PCR method for quantification. So, okay, through 2 rounds of amplification. So this is a schematic showing the 2 rounds of PCRs that I mentioned earlier. So we do a first round PCR with a sample cDNA or DNA, and typically we'll put in 6,000 molecules of the internal standard molecule for the different genes in a mixture. There'll be a primer mixture for each of the targets that we want to assess. And at this point, there are no probes. We go through 18 cycles to amplify it. So this is just showing sample 1 here in one well of a microplate, and showing that for the different targets here, genes A, B, C, and D, which are different levels in the cDNA, they're being measured relative to a set amount of internal standard molecules, in this case 2 molecules for each internal standard. So for this gene, the ratio would be 4 to 2. For this gene, 2 to 2, this one 1 to 2, and this one 2 to 2. So then this product from 18 rounds is then diluted 1,000-fold and dispersed into multiple wells in a microplate. And in the second round, then, there are, in each of these wells, our primers for a single target, and we then put in probes for the FAM for the native template and the Quasar 670, the internal standard for the one target. And then we amplify that 40 cycles. And so in each of these different wells with a different primer pair and different probe, you'll then be able to see the PCR reaction signal and at the different ratios, and be able to quantify how much you have there based on the NT-CQ compared to IS-CQ. And so in the next slide, then, this is how we set it up in a 96-well plate. This is set up for 20 samples, measuring 4 different targets in each of these 20 samples. So each of the samples is mixed in a first round amplification with an aliquot of internal standard. It's pre-amplified through 18 rounds, then diluted 1,000-fold and put in the individual well. We then measure each gene relative to this known number of internal standard molecules. We're measuring 4 genes per sample, so we've got 4 rows here. Each column is a different sample. Each row is a different target. And we have 2 different sets of these. So this is the first set of 10 samples. And then down here we have the second set of 10, so 20 samples. We then measure each gene also in an external standard mixture at 2 different concentrations, which are represented in these 2 columns. And we also have no template controls down here to ensure the absence of false positive results there. So in sample 1 then, in this well, in this set of wells, we would have the cDNA, the internal standard mixture. The internal standard mixture would have possibly different internal standard concentrations, 600,000 molecules for beta-actin, 60,000 for mix, 6,000 for E12-1, and 6,000 for E21. We have sequence-specific probes, FAM for the NT, Quasar for the internal standard, buffer, CNTPs, and the TAC enzyme. So that's then for the external standard mixture, we have these 2 that we expect, and if there's any variation from experiment to experiment for this expected, we use that as the correction factor from experiment to experiment for the values for the actual samples. So this is an example of the LTD performance in FFPE samples, surgical FFPE, and the cutoff value, which was derived from this ROC curve, had a 90% specificity, 90% sensitivity, with an ROC curve POC of 0.93 and a p-value of 0.0061 for stratification of malignant from non-malignant. Now, when we applied this then to the FNA samples, the performance was not quite as good, mainly because of the quality of the FNA samples, which I'll go over the next couple of slides. But the p-value, again, was very good. The cutoff value, in this case, had a 92% specificity and 75% sensitivity, and the ROC curve AOC was 0.87. So if we just look at the types of samples we have for FFPE, we have a very large piece of sample surgically, which is typical, but we don't get surgical samples for most lung cancer cases. But these are very good. If you look at the analysis on the adjuvant, this is the kind of curve you'll get. And for FNA FFPE samples, in many cases we get a good adjuvant curve, but you can see there's much less sample here. And so there is, for many of the samples, you will not see that. And we found that when the RNA yield is less than 150 nanograms, because of the low RT efficiency and then the low PCR efficiency due to the FFPE treatment, we wind up with a failure oftentimes for these samples. So that winds up being the big issue. So we've identified methods for optimizing signal. We have optimized our RNA extraction method. We have also optimized reverse transcription conditions, and then we use the multiplex 2-round PCR to be able to assess as many of these FNA samples as possible. One of the advantages of we found for RT is that if we use a gene-specific RT as opposed to oligo-DT analysis, then we get a much greater yield. In this case, we're looking at beta-actin. You can see for these 3 different samples, this is a malignant sample 1 and 2, and then benign sample 1 here. You can see that the yield of cDNA is 2 to 3 logs better if you use the gene-specific priming. So that's a key feature in order to improve signal. And this is loaded. You can see the amount of RNA loaded here. So the in a prospective trial. We plan to assess hundreds of transthoracic FNA cell block FFPE samples. We'll compare the LCDT analysis to the cytomorphic analysis and compare sensitivity and specificity, and assess the ability of the LCDT not to replace, but to augment the accuracy of cytomorphology. So next, I'm just going to talk briefly about this test for viral hemorrhagic septicemia, and it was in this paper by Pierce et al. Last year. There was a need for a faster, more accurate diagnostic test. We developed a test with the fish beta-actin gene as the loading control. We had internal standards for the viral hemorrhagic septicemia N gene and the beta-actin 1 gene. We used a SAM BHQplus probe for BHSV, beta-actin NT, and a Quasar 670 probe for each IS, both for the internal standard for the BHSV and for the beta-actin. We demonstrated a high signal-to-analyte response with a very good slope and the linear dynamic range that was again over 7 orders of magnitude. And the conclusion was that this assay is a very rapid and inexpensive, has significantly greater accuracy than other published tests and traditional cell culture methods. And so we are now beginning to apply that in tests of BHSV from Lake Erie and other Great Lakes samples. So, and that's what I just mentioned here, so we're evaluating that in the largest study. So the summary then is that for most, for more robust measurement and highly degraded FFPE and FNA samples, we use a competitive template mixture of internal standards from Genomics Incorporated in combination with sequence-specific fluor-labeled hydrolysis probes from BioSearch Technologies. And this controls for interfering substances that prevents false negative results, enables implementation on the commonly available real-time thermocycler devices, enables use, of course, of short PCR products, which is important for degraded samples. And so so this is the information for Accurate Genomics, and this is the acknowledgement for our lab, our group here, and also our collaborators here. So I think that's it, Andrew, for my presentation. Thank you for the attention.
Great, excellent. Thank you for that. We'll next be hearing from Cassie Temple. Cassie.
Thank you for the introduction, Andrew. And today I'm going to be talking about BHQplus probes and the ArrayTape platform, a successful combination for accurate and economical SNP genotyping. I'm going to start with a presentation outline. First, we're going to look at a SNP genotyping overview, then review some methods for SNP analysis. We're going to look at BHQplus SNP genotyping assays in a little more detail with an introduction to the ArrayTape platform, and then we will review some case studies with BHQplus probes and ArrayTape, looking specifically at human, corn, and bovine examples, and then summary and conclusions. So starting with a SNP genotyping overview, SNPs are single nucleotide polymorphisms found in the sequence of genomic DNA that is otherwise identical. And at the bottom of the screen I have a Sanger sequencing result. In the center you can see there are really 2 nucleotides sharing one position. This would be an example of a heterozygous SNP. SNPs are genomic DNA sequence variants between members of a biological species or paired chromosomes of an individual. And by that I mean you can see differences globally. For example, corn that grows well in Brazil would have a very different SNP panel than corn that grows well in Wisconsin, or also one individual could inherit a C allele from mom and a T allele from dad at the same location. Alternate forms of genes or SNPs are called alleles, and we'll be using that term quite a bit today. So some SNP genotyping applications include clinical diagnostics, feed animal genetics and production, as well as crop development such as corn and soybeans. For human research and diagnostics, SNPs are used widely in disease prediction as well as pharmaceutical drug response prediction. So SNPs are very informative to doctors as they're making decisions about which pharmaceutical drug to prescribe and which dose to prescribe to a specific individual, as well as genetic research. Ag biotech research and development uses SNPs widely for marker-assisted selection programs. SNPs are also widely used for backcrossing of plants and animals, as well as trait associations, and we'll be looking at an example of that later, and new crop and development. SNP genotyping methods. We're going to start with some non-PCR-based SNP genotyping methods. Of course, the first one we think of is DNA sequencing. With whole genome sequencing, the entire genome of one individual would be sequenced, so therefore all of the SNPs would be known. Also, targeted genotyping by sequencing, known as GBS, is used, and in this case, only a specific region of interest would be sequenced to get the information that's needed for a specific study. Microarray technologies, such as those available from Affymetrix or Illumina or Exaquan, are widely used, and these usually have a kind of a set panel of SNPs that can be used to analyze hundreds or thousands of SNPs on an individual sample. There are several technologies available for SNP genotyping with real-time PCR instrumentation. Melt Doctor and Cybergreen reagents are used to distinguish SNP alleles with melt curve analysis after PCR, so the PCR itself does not need to be completed on a real-time PCR instrument, but the analysis does. So the example I have on the right side of the screen shows 3 different melt curve profiles of samples, and this can be used to distinguish the SNP alleles of those samples. Molecular beacons distinguish alleles in real time during the PCR reaction, and because of the specific temperature at which beacons hybridize to the sample specifically based on the SNP allele, the fluorescence detection needs to occur at a controlled temperature. There are several technologies available for SNP genotyping with endpoint PCR, and all 3 of these technologies that we'll talk about allow researchers to complete the PCR reaction and then scan the fluorescence results at room temperature at a later time. First, we'll talk about competitive allele-specific PCR, also known as CASP assays. These are from LGC Genomics, and this is a FRET-based technology. TACMAN assays from Life Technologies are hydrolysis probe-based with a minor groove binder, and these are also referred to as MGB probe assays. BHQplus assays from Biosearch Technologies are hydrolysis probe-based assays with duplex stabilizing technology and black hole quenchers. And on the right side of the screen I have an example from each of these types of assays. We looked at a set of samples with the same SNP using all 3 of these technologies, and as you can see, the results are very similar for all 3. From here on out, we will be focusing our attention on the BHQplus assays from Biosearch Technologies. So as a lab is deciding which SNP genotyping technology to use, there are a couple of things that need to be considered in making that decision. First would be the sample preparation protocol. For example, the cost per sample, speed and ease of use of that protocol, and compatibility with the assay and master mix of interest need to be considered. The assay design, whether it's to purchase a pre-designed, pre-validated assay set or design custom assays, is part of the decision as well. And the reaction volume of the PCR reactions that are going to be completed. This is going to be to a large degree dependent upon the platform of use. Of course, the cost of master mix and the cost of assay are going to play a role as well. So we're going to take a closer look at SNP genotyping assays with BHQplus probes. Some distinguishing features of these probes and genotyping assays are that BHQplus probes form highly stable duplexes with DNA targets, and this is important because it allows for shorter probes with enhanced target specificity, and that really gives the user much more leeway in how they design their assays and where they place those primers and probes. And this also makes it ideal for SNP discrimination because you do need that highly stable duplex with very specific annealing to get proper SNP discrimination. The black hole quenchers do not interfere with fluorescent dye signal after hydrolysis. So this is important for getting those nice clear cluster plots at the end. And BHQplus assays are easy to design and order with the real-time design software available on the Biosearch Technologies website. And all of the examples that we're going to be sharing today were designed with that software on their website. Importantly, also, BHQplus probes provide a cost-effective method for SNP genotyping with hydrolysis probe-based assays. So just to give an overview of what a BHQplus SNP genotyping assay consists of, each of these assays consists of really 4 components. You have a forward primer, a reverse primer, and an allele-specific probe set. In this example, we have a genomic DNA strand with a C to A SNP in the middle. The probe that binds specifically to the C allele is labeled with a FAM dye, and the probe that binds specifically to the A allele is labeled with a CalFluor orange dye. Both of these probes have a black hole quencher attached to them. So during the PCR process, this is kind of an image in the middle of a PCR process during the annealing step after the double-stranded genomic DNA has been denatured and single-stranded. The primers and allele-specific probes will anneal. So the forward and reverse primers bind to the outside of the SNP location indiscriminately of the allele in the center, but the probes will bind specifically to the allele that they are targeting. So in this case, the FAM-labeled probe will bind specifically to the C allele, and the CalFluor orange labeled probe will bind to the A allele. In this conformation, both the FAM and CalFluor orange dyes are still quenched because they are held in close proximity to that black hole quencher. As PCR progresses and Taq polymerase extends, the allele-specific probes are hydrolyzed by exonuclease activity. So as the Taq enzyme moves along and creates that second strand, it will encounter the probe and hydrolyze it. In the process, it essentially breaks it into small pieces and separates the quencher from the dye. In this case, a C allele would result in a FAM dye being unquenched, and the A allele would result in a CalFluor orange being unquenched. And this is what connects the dye color to the genomic SNP allele. So here we have an example of a SNP genotyping cluster. So this is what the end result would look like after a SNP genotyping reaction is complete. Up here on the upper left, we have the CalFluor orange only cluster. This would represent the homozygous A alleles. In the middle, we have FAM and CalFluor orange fluorescent dyes. This represents the heterozygous A and C alleles. And down here, we have the FAM only cluster, and this is the homozygous C allele. So the one that I showed you before was what we want all of our clusters to look like, but in reality we get cluster plots that are in kind of a fairly wide variety of shapes and sizes. Here we have some clusters that have the heterozygous cluster closer to the FAM side, and then sometimes the heterozygous clusters closer to the Vic side. Some of the clusters can be large and spread out, and sometimes they're a little bit more elongated. However, in each of these cases, the clusters themselves can be scored with confidence. So now I'm going to switch gears a little bit and talk more about the ArrayTape platform. So the ArrayTape platform consists of 4 major components: ArrayTape itself, the Nexar, the Solex, and the Arraya. ArrayTape is a flexible microplate replacement, and as most microplates are kind of stiff polypropylene thick plates, ArrayTape is a very thin polypropylene that's flexible and all held together in one long reel. This allows for reduced reaction volumes. Our standard tape comes with a total reaction volume of about 2 microliters, and this is sealed with an optically clear seal, which allows the fluorescent dyes to be detected through the seal. The Nexar is the liquid handler that's optimized for use with ArrayTape. In a standard reaction, we would run a total volume of 1.6 microliters, so the Nexar would dispense 800 nanoliters of DNA with 384 samples being dispensed at one time. And we would also dispense 800 nanoliters of mass mix into 384 wells within 38 seconds. The Nexar then seals the ArrayTape and prepares it for thermal cycling. So thermal cycling occurs in the Solex. This is our high-capacity water bath, and one Solex can hold up to 600 384 well arrays at one time. The Solex is optimized for use with ArrayTape, and it has 3 individual tanks that are used for PCR protocol optimization. Touchdown, 2-step, and 3-step PCR protocols can all be used in the Solex. The Arraya is our endpoint fluorescent scanner. It has 3 channels. The first is the FAM channel, the second detects Hex, Vic, or CalFluor orange, and the third channel is for rocks, or Texas red, I guess. The Arraya scans 384 wells within 28 seconds, and after scanning in the Arraya, the data is ready for analysis in the Douglas Scientific Intellic software or other analysis software. So just to review the workflow with the ArrayTape platform, we start with the ArrayTape, which is our microplate replacement. We then will go on to the Nexar for our liquid handling. Thermal cycling occurs in the Solex, and fluorescence detection is completed in the Arraya. So even though the instruments we just talked about are very different from what you might see in a standard laboratory setting with a real-time PCR instrument, the results are surprisingly similar. So here we have a picture of a cluster plot that was generated in the array tape platform with 1.6 microliter reactions, and a matching set of samples and the same assay run in a real-time PCR instrument with 5 microliter reactions. And as you can see, the results are ultimately very much the same. So now we're going to move over to some SNP genotyping case studies where we use BHQplus assays and the array tape platform. And we're going to start with a human SNP genotyping experiment. For this study, we started with human genomic DNA samples, 34 purified DNA samples from cell lines that we purchased from Coriell cell repositories, and these samples were highly purified and diluted to 5 nanograms per microliter in water before use. The SNP genotyping assays that we used for this experiment were 2 different sources. We started with pre-designed TaqMan SNP genotyping assays from Life Technologies, and we also used custom BHQplus SNP genotyping assays designed with that real-time design software. For this study, we chose our SNPs for their pharmacogenetic significance. All of our reactions were completed with TaqMan genotyping master mix from Life Technologies, and we used 2 different instruments for this study. The VS7 from Life Technologies with the 384 well plates and 5 microliter reactions, and also the array tape platform with 1.6 microliter reactions. So a little bit of background information on these SNPs that we used. VCOR-C1 and CYP4F2 are both known to be important for metabolism and dosage of warfarin, which is a very commonly prescribed medication. KIF6 is known to be important for coronary heart disease. And the reason we chose these SNPs for this study is because the consensus genotype calls for those 34 Coriell genomic DNA samples were publicly available, so we could use these to compare the performance of our instrument and the custom assays to the known published results. So here are the cluster plot results for VCOR-C1. On the left, we have the TaqMan assay with a VS7 real-time PCR instrument. So this is kind of what you would expect to see in a lot of standard laboratory setups. This was a 5 microliter reaction with a real-time PCR instrument and a pre-designed TaqMan assay. We then moved the TaqMan assays into the array tape platform and got a very similar result. And then we also moved over to the BHQplus assays in the array tape platform and again had a very, very similar result. So when we scored all of these results, we were able to compare the published or expected genotype with the genotype that we scored from the TaqMan assay with the VS7, the TaqMan assay with the array tape platform, and the BHQplus assay with the array tape platform. And all 3 of these conditions match the expected calls for all 34 cell lines. So moving on to CYP4F2, again we have the real-time PCR instrument with TaqMan assay. On the left, the TaqMan assays with array tape platform in the center and BHQplus assays in array tape on the right. Same as before, we have 3 nice scoreable clusters and very similar performance with all 3 conditions. And here are the results for this. All 3 of the conditions that we ran compared very well and matched exactly to the expected or published results. And KIF6, here's the 3rd assay that we were looking at. Again, we have all 3 conditions, and you can see on the right side, the BHQplus assay, those clusters were a little bit closer than before, but when we scored them, everything matched the expected, and we were able to confidently score both of those 2 clusters. So our summary and conclusions from this study was that custom BHQplus SNP genotyping assays produced the expected calls for each sample and assay combination. They performed equivalently to the pre-designed TaqMan assays, and they were successfully miniaturized to a 1.6 microliter reaction. Our conclusions for the array tape platform were that it produced expected calls for each sample and assay. It performed equivalently to the real-time PCR instrument, and overall the combination of BHQplus SNP genotyping assays and the array tape platform matches the performance of pre-designed reagents and the real-time PCR instrument. So next we moved on to a corn SNP genotyping experiment. And this experiment was a little bit different. We started with 3 SNPs on chromosomes 1, 3, and 4 of the corn genome. And these SNPs were identified to be highly polymorphic by Mamedov et al. And we chose these SNPs not because they were necessarily associated with any specific traits in corn, but because they were polymorphic and we could use them to really demonstrate the assay and sample prep optimization and how different things impact the performance. So we will refer to these as SNP 115, 176, and 611, which is the last 3 digits of that SNP ID. So a little review of the reagents that we used here. We started with corn samples. We had yellow field corn samples that were obtained from 11 different sources in central Minnesota, and this included local farmers and grain elevators. We took 8 individual kernels from each of those sources for a total of 88 individual samples, and we produced crude preps for each of these individual kernels using a very basic sodium hydroxide crude prep method. And this is...
We have lost Cassie's audio there. At least I can't hear anything at this point. So please stand by. If you have any questions for Cassie, please write in. I'm just going to give her a quick message here.
Did I just need it? Can you hear me?
Can hear you. Sorry, just
okay.
Audio dropped there for a moment.
Okay.
Thanks.
All right, I'll keep going. Sorry, so the crude preps that we use here were very different from the highly purified DNA samples from the previous experiment for human samples. And for this experiment, we used only custom BHQplus SNP genotyping assays from Biosearch Technologies. And as I mentioned earlier, the SNPs were chosen because of their polymorphic nature. In this experiment, we looked at 4 different master mixes. So ClearCall master mix from LGC Genomics, TechMan GT Express master mix from Life Technologies, Perfecta qPCR Tough Mix from Quanta Biosciences, and SSO Advanced Universal Probe Super Mix from Bio-Rad Laboratories. And we chose these 4 master mixes because we expected them to work well with these crude preps. Again, we use a VIA7 real-time PCR instrument with 5 microliter reactions, and the array tape platform with 1.6 microliter reactions for this study. So here are the cluster plots for SNP 115. You have very similar cluster plots for all 4 of the master mixes, including ClearCall, the upper left, GT Express on the upper right, Perfecta, and SSO Advanced. So all 3 of these produced easily scoreable clusters and demonstrated that highly polymorphic nature of these SNPs. So our summary for SNP 115, all 4 of the master mixes produced the exact same call for the exact same sample, and the array tape platform calls matched what we saw on the VIA7, and in this case all of our calls were made, so 100% call rate and 100% concordance across the conditions. For SNP 176, again we saw 3 easily scoreable clusters for all 4 of the master mixes, and again our results were all concordant with each other across master mixes and platforms with, again, 100% call rate. For SNP 611, we have, again, 3 highly scoreable clusters with all 4 of our master mixes. This SNP was not quite as polymorphic as the other ones, but still produced the 3 easily scored clusters. And once again, we had 100% concordance across the platforms and the master mixes with a great call rate as well. So our conclusion from this study was that crude DNA samples are compatible with BHQplus assays and the array tape platform, that SNP genotyping calls were consistent across multiple master mixes and across multiple instruments. The call rates in this case were 100% for each assay, although I wouldn't promise that for every single assay. The SNP genotyping calls matched the array tape platform and the VIA7. So therefore, when you combine all of this, the inexpensive crude prep protocols and miniaturized reactions enable incredible cost savings for SNP genotyping analysis of agricultural samples. So next we'll move on to our bovine SNP genotyping experiment. This experiment, we started with 29 beef samples that were obtained from a variety of grocery stores in the Minneapolis, Minnesota area. For this experiment, we looked at 3 different sample preparation protocols, including a purified DNA sample that we purified with the QIAGEN DNeasy blood and tissue kit. We had 2 crude prep protocols. One was the Quanta Biosciences Extracta DNA prep kit, and also a sodium hydroxide crude prep protocol that we found in the literature. For this, our SNP genotyping assays were all BHQplus assays designed with the real-time design software, and in this case, we chose our SNPs based on their commercial importance to the beef industry, and we'll go into that in the next slide. Also for this one, we use 6 different master mixes to look at how these sample prep protocols and the master mixes interacted with each other. So here we had TaqMan GT Express and TaqMan Genotyping master mixes from Life Technologies, Perfecta qPCR Tough Mix from Quanta Biosciences, SensiFast Genotyping and SensiMix probes from BioLine, and SSO Advanced Universal probes from BioRed Laboratories. In this case, we only looked at the samples in the ArrayTape platform with 1.6 microliter reactions. So to give a little background on the SNPs that we analyzed, the first one that we looked at was the leptin gene, and this SNP is associated with body fat in cattle. The homozygous C allele is associated with lean cattle and is not economically favorable. The heterozygous CT allele is economically intermediate, and the homozygous TT allele is associated with fatter cattle and is economically favorable. CAPN1 4751 is a SNP that's it's associated with meat tenderness. The homozygous T allele is less tender and not as favorable. The CT heterozygous allele is intermediate, and the homozygous C allele is favorable for tenderness. UGCast is the third SNP that we looked at in this study, and this SNP is also linked to meat tenderness. The homozygous G allele is less tender and not favorable. The heterozygous CG allele is also less tender and not favorable, but that's because the G allele is dominant. And the homozygous C allele is the most tender and therefore favorable. So the first thing that we did is we looked at our DNA sample preparation comparison. In this example, we have CAPN1 4751 SNP assay. In the columns from left to right, we have purified DNA, the extracted kit, and the sodium hydroxide crude prep. And in the top row, we have TaqMan GT Express master mix. And in the bottom row, we have TaqMan Genotyping master mix. So this is the exact same sample set with the exact same assay. But as you see in the top row, we had 3 very nice, easily scored clusters. But in the bottom row, we had only scorable clusters for the purified DNA and essentially no amplification for the crude preps. So we looked at this a little bit farther with our leptin SNP assay. Here we had a master mix comparison looking at the sodium hydroxide crude prep. So when we looked at all of the master mixes in our study with the sodium hydroxide crude prep, about half of them produced easily scorable clusters and about half of them did not. So for the rest of this study, we focused on the purified DNA. So this is, again, the exact same assay, exact same sample set with purified DNA, and all 6 master mixes were very easily scored. For CAPN1 4751 SNP assay, the master mix comparison, again, all 6 master mixes showed very nice, easily scored clusters for this SNP. And the UGCast SNP, again, all 6 master mixes worked very, very well. So a summary of the bovine SNP genotyping results. I have a box on the right where I organize them from most economically favorable to least based on the expected genotypes there. Unfortunately, none of our samples had the ideal combination of SNP alleles, but 31% of the samples did contain 2 of the 3 ideal alleles, so that was pretty good. Interestingly to us, several of the packages contained both highly favorable and highly unfavorable samples. So this was when we purchased the meat, we would purchase a package and number the package, but then assign a letter to the actual piece of meat. So if you look at sample, sorry, let me get my mouse, 1B, it's way up here at the top, but sample 1A is down here. The same thing happened with package number 3, where 3A was at the top and 3 was way down here. So that was very surprising to us to see. Also, we were very interested to see the impact of the sample preparation method and the master mix, that combination must be compatible to get the results that you want. So from here, we'll go to overall summary and conclusions. And we've talked a little bit about the cost considerations here and there. While we were putting together these experiments, I kept track of the catalog pricing of each of the components that we used. For some of the human assays that we analyzed, they were part of the clinical panel from the TaqMan assay clinical panel, and some were in the TaqMan standard assay panel. And we also compared to custom BHQplus assays. And the upper part I have 5 microliter reaction volume, and the lower part is 1.6 microliter reactions with the different master mixes in between. So when we looked at the catalog pricing, we set GT Express with a TACMAN clinical panel and a 5 microliter reaction at 100% of the cost and scaled everything accordingly. So just looking at the top row, you can see that if the only thing that changes is the source of the assay, you can save between really 65 and 75% of the cost. And if you go all the way down to that lower right section there, you can save between 89 and 92% of your reagent costs. And again, this is based on catalog pricing. So overall summary and conclusions were that BHQplus probes are effective and accurate for SNP genotyping, and with a wide variety of samples. We showed it with purified DNA, anywhere from the highly purified cell lines to very crude corn and crude beef samples. The ArrayTape platform allows researchers to miniaturize and automate PCR analyses such as SNP genotyping with accuracy and high call rates. And overall, the combination of BHQplus probes and ArrayTape enables accurate and reliable SNP genotyping results while providing significant cost savings. So with that, I would like to thank you so much for your time and attention.
Great. Well, thank you so much for that, Cassie. That was a great presentation, and thank you also to Dr. Wiley for his portion of the presentation. At this point, I would like to invite our audience to continue sending their questions or comments in right now using the questions window for the Q&A portion of the webinar. And I've already received some questions here, so I'll start with the ones that we have. Our first question: how many targets can multiplex reliably?
So yeah, this is. Can you hear me, Andrew?
Yeah, we can hear you. Thanks.
Okay, so this is Jim Willie. So yes, it's possible. We have multiplexed hundreds of samples, hundreds of targets. Excuse me. It is important to use a design program such as that available through NIST to ensure that primers are compatible and will not form robust primer dimers. With that, as long as they're carefully chosen, one can get to over 100, and some have described hundreds of multiplexing hundreds of targets at the same time. And so that is certainly possible, and we have done that ourselves. And we've reported that going back to a paper by Crawford et al. In early 2000. So, thank you.
Excellent. Thank you for that. Our next question here: how do SNP genotyping results in ArrayTape compare to SNP genotyping results in microplates exactly?
It compares very well. Generally, we, for the most part, can take a fully functional SNP genotyping assay from a microplate and transfer it right into ArrayTape with very little optimization. Sometimes we do see some differences in primer binding, and so we might need to change a little bit of the PCR conditions, or in some rare cases we would have to go back and redesign the primer or probe sequences, but generally it will transfer very nicely.
Excellent. Okay, thank you for that. Our next question here: can you discuss the advantages of running 2 amplifications rather than just using the individual probes and primers for each marker in a single round of amplification?
Yes. As people may understand, there are limits to how many fluorescent signals one can reliably sort out with the current software and photomultiplier tubes and so on. So I think the maximum I've seen is about 10 different fluors that have been sorted out, but the problem increases significantly as one adds fluorescent probes. So that's why for looking at dozens or hundreds of products, it's probably going to be better to sort them out the way I described, do an initial multiplex, and then do a second round of amplification. Do the first round with multiplex with internal standards, so you can fix that quantification, and then dilute and put in a second round with a smaller number of primer pairs and probes.
Okay, thank you for that. Our next question here: Can you please discuss how the quality cluster plots are generated? So this question came in during, Cassie, your core and Snip examples. What were the axes exactly there?
Oh, sorry. Let me explain that a little bit more. So the axes are, in this case, were normalized fluorescence dyes. So the y-axis that we were showing was the signal of CalFluor orange divided by rocks. So that was our normalized CalFluor orange signal on the y-axis, and the x-axis was the FAM signal divided by the rock signal. So a normalized dye on the x-axis as well.
All right, excellent. Thank you for that. Our next question here, what are the different sources of a suboptimal PCR reaction that competitive PCR controls for?
Yeah, that's a really good question, which I alluded to some degree. But some of the problems are that each sample may have different amounts of inhibiting substance interfering with the PCR efficiency. And those interfering substances may affect some targets more than others. So if you have an internal standard that for each target that is affected the same way as the native template, then you can control for that inter-target variation in interference. So that's one thing. Another is that there may be variation from well to well within a plate. In terms of the efficiency, the thermal cycler itself may not have perfect efficiency from one well to another. And in addition, if one is using a multi-pipetter, then if it's not working perfectly, one may get different amounts of reagents loaded from one well to the next, and that can affect the efficiency, which the internal standard will control for that as well. So those are a few topics.
Excellent. Okay, thank you for that. Our next question, do you ever encounter data quality problems when miniaturizing reactions from microplates to array tape?
Generally, no, we don't expect to see problems. Sometimes, as I alluded to earlier, we will see some primer binding differences, and this has more to do with the thermal properties of that very, very thin array tape well size. So the tape is very thin, and when you're coming from very thick microplates with a Peltier block, and then you're moving it over to very thin, small volumes in array tape, in a water bath PCR, the annealing conditions are a little bit different. So sometimes we have to account for that, but generally there's not a difference in data quality.
Right. Okay. Thank you for that. And this one perhaps for both speakers, if you can comment. Could you give some input on your experience designing the assays? Does this present additional challenges to assay development?
Well, there certainly are design considerations. There are limitations. It depends on how specific the target you're setting is. If there are limitations, for example, in measuring transcript abundance, where typically you want to cross an intron to minimize the effects of genomic DNA contamination, then that can limit to some degree. And if there are suboptimal sequences for priming due to GC-rich or AT-rich areas, then that can have an effect, especially if you're trying to develop a short product like 60 to 80 base pairs, which is our goal in these situations for assessing fragmented material, then that can be a factor. And if you have a very short gene, then that can be limited as well. I think Cassie can also comment, because when you are assessing polymorphic sites, obviously you're very limited to those regions. So.
Yes.
Right.
So
go ahead.
I would say overall my experience with designing these assays has been very positive. We've been able to use the real-time design software with incredible success. When we initially tested this, we went a little bit outside of the rules, I guess, and designed multiple forward and reverse primers just to make sure that we got the best possible combination. But ultimately what turned out was that the I guess, suggested primers from the real-time design software actually worked very, very well. And as terms of does it present additional challenges to the assay development, I wouldn't necessarily say that it does always, because sometimes you really want to have that control over the final design of the assay rather than purchasing pre-designed assays where you don't know exactly where the primers are. You know the region, but not precisely where they are. So if you're in a highly polymorphic region or a area that could be duplicated in multiple areas of the genome, you want to know exactly where those primers are sitting and have complete control over that specificity. So for me, it was kind of a positive to know exactly what was going on with those primers.
Yeah, one thing I would point out, Andrew, is that no matter what in silico methods you use for designing primers, they really do need to be validated through experimentation. And typically, probably 20% of the primers you design will be suboptimal, depending on what level of efficiency you're looking for. So, something to consider.
Interesting. Okay, thank you for that. Another question here. Is the array tape platform reaction volume set at 1.6 microliters, or can other reaction volumes be used?
Other reaction volumes can be used in the array tape platform. Many of our customers actually dry down their DNA samples and run in a total reaction volume of 800 nanoliters or even as low as 600 nanoliters. So 1.6 microliters is kind of actually the upper limit for our standard array tape, and so many customers go down. However, we do have custom tape formulations that can be used to go higher than 1.6 as well.
Okay, excellent. Thanks for that. Another question. Since cluster plots can have different shapes, how can you be confident in your calls? Do you often rerun samples, or how does that process work?
Usually we don't rerun samples. As I showed that one example with multiple different kind of personalities of cluster plots, as you work with SNP genotyping assays, the more you get to know them, the more you kind of know from day to day what to expect from a specific SNP genotyping assay. Generally, the cluster plots are very easy to score, but depending on who you are, I guess if you're more aggressive or more lenient, then you might score things as being for sure, or you can always leave things as indeterminate between the clusters if you're not certain of what the actual sample genotype is.
Okay, thank you. I think one last question here. How many SNP genotyping reactions are actually run in array tape, and how many instruments are in the field right now, do you think?
We have about 150 instruments globally deployed right now, and we estimate that about 1 to 1.5 billion SNP genotyping reactions are run in array tape every year.
All right. Well, thank you both very much for that, for all those answers. We have reached the end of the question and answer portion of this webinar. If you do have any further questions, however, you can direct them to the email addresses showing on your screen right now. That's james.willie2@utoledo.edu. Or for Cassie Temple, you can write her at cassie.temple@douglasscientific.com. Questions for BHQplus probes can be directed to tech support at biosearchtech.com. Thank you, everyone, for participating in today's conference. You'll be receiving a follow-up email from X-Talks with access to the recorded archive for this event, and a survey window will be popping up on your screen. Your participation is appreciated, as it'll help us improve our further webinars. Now, please join us in thanking our speakers today, Dr. James Willie and Cassie Temple. We hope that you found this conference informative. Have a great day, everyone.